Hey there, I just wanted to show you how to fill in the cell transport review activity that we are doing today on Wednesday. So I'm going to click here to download a copy. So I'm going to click make a copy. Um, excuse me. Okay. And then you should have a Google slide that opens up. And then I'm going to zoom in, make this a little bit bigger. So I'm going to make sure to put my name up here at the top. Okay. That way, my name's on my paper. And then here in part one, they want you to complete the table by checking the correct column for each statement. I included little stars for you instead of little check marks. So basically, you'll just drag these stars to wherever they go. Okay, so doesn't change the shape of the cell, causes the cell to swell. Okay, so you'll just figure out where these need to go. And obviously, you can use your notes, absolutely. So there should be enough stars for you to fill in the table. Part two, match the term with its correct description. So again, is used during active transport, but not during passive transport. So you'll come over here and you'll type in, is it osmosis? Is it exocytosis? Please actually type out the answer. Don't just put A. I mean, you can put A osmosis, but it should say, E facilitated diffusion, right? Whatever the answer is. So you'll just type that out. And then down here, part three, label the diagrams of cells using the following terms. And then I gave you the terms, so you can just click and drag these terms to wherever they belong for these 6 cells. So that's page 1. Page 2 up here at the top, match the structure or process to the letter. So phospholipid bilayer, right? Which letter would you grab? And this is the image that you're looking at, and it should look very familiar. It's, the same image from your test. And then the next few questions, you're basically just selecting the best answer. So what I did is I gave you a box. So this cell would be in a, is it a hypertonic? And if it is, you'd put the box around it. If you think it's hypotonic, you put the box around that. Or isotonic, you'd place the box around that one. Okay, so you're just moving the boxes to decide which is the best answer. Okay, so you do that for 6, 7, 8, 9, and 10. Part 5, in the diagram below, draw the arrow that water will move in each of the beakers. So I gave you the arrow. All you have to do is manipulate it. So if you think water is going to move into the cell, then obviously we would put the arrow like this. And I can make it smaller, right? I can make my arrow a little bit smaller. So going in, if you think water is going to come out, then it would look like this. Okay. So you'll just manipulate the arrow. Where do you think water's moving? And then we need to, so it says in the diagram below, draw the direction. And then they want you to label it as either hypotonic, hypertonic, or isotonic, I believe. Yeah. So, right, this one has 10% salt on the inside of the cell, 10% salt on the outside, which means I'm going to have 90% water inside and outside. So I would label this as isotonic. Yeah, so in the diagram below, label each beaker. Okay, so that's what I would do there is I'm going to label them hyper, hypo, or isotonic. Page 3, part 6 is still looking at osmosis. So you're filling in the blank. So I think this goes to cell A. So water will flow, and then you're going to type, is it into the cell, out of the cell, or in both directions? And then what's going to happen to the cell? Is it going to shrink, burst, or stay the same? So 1A and B, 2A and B, and then 3A and B. Okay, all on osmosis again. Part 7 is looking at active transport. So you have an amoeba that has engulfed a particle of food. Does it require energy? Is it active or passive? And then is it endo or exocytosis? And then in this picture, it's the same thing. It's still an amoeba. So this is active transport. So that's a good hint for you on these 2 questions for part 7. And then finally, page 4. At which solution of concentration gradient is each cell diagram? So again, hypotonic, hypertonic, or isotonic. So that's what you would type here, right? Hypotonic, hypertonic, or isotonic, okay? So it's going to be one or the other, okay? So if you label this one, for example, if you label this one hypotonic, then obviously this one cannot also be hypotonic. Okay, so you'll use each of those terms just once. Is this diagram is moving from high to low concentration, so is that going to be active or passive transport? Using a transport protein to move particles across the membrane, what type of transport is that? If I am using a transport protein to move particles, it looks like I'm moving from an area of high to an area of low. So that's a good hint on what our answer right here will be. And then describe the processes occurring in the following pictures. So, this amoeba, oh no, it's engulfing something. So what type of transport is that? And then this middle one, right? You're starting here, number 1, with this vesicle, and then 2, and then 3. So this cell is getting rid of something. So this cell is eating, it's engulfing. This cell is removing particles. And then in this image, we're going from a low to a high concentration. Okay, so how will we label these 3? So that's it. These 4 pages just to review active and passive transport. If you have any questions, just let me know. Thanks.